H's. What a remarkable idea of evolution. Mothers are literally recruiting another life form to babysit their baby. And they're making sure that this particular bacterium flourishes in the baby because that's the one they feed. So, what do we learn from this? Well, we're not alone. The bacteria in us is clearly very important to our health. From an evolutionary perspective, there's more human milk, oligosaccharides, in milk than there is protein. What that means is it's basically as important to feed the bacteria in the baby as the baby. So we're beginning to realize that the bacteria in us is far more than simply a supply of calories. It's protecting us from pathogens. It's educating immunity. It's educating metabolism. It's providing a crosstalk between the host and the microbial world. And we're learning what that microbial world is like by this interaction. If you get the bacteria right, you get fuel, you get vitamins, you get protection. If you get bacteria wrong, you get the exact opposite, endotoxin and toxicity. Now, this is already an industrial practice. That is probiotics. People consume live bacteria, and in essence, they think that that's affecting the bacteria in them. And the truth is, this generation of probiotics does not do that at all. Probiotics go in and probiotics come out. So probiotics are basically just living bacteria that go through you. What this bacteria does in babies is quite different. It multiplies. So when a baby gets this bacterium in its intestine, the bacteria multiplies and multiplies and multiplies. And in fact, if a baby is breastfed and has this bacterial strain, over 90% of the weight of the diaper is a single strain of bacteria. And it's clearly protecting the baby, educating immunity, protecting it from all sorts of problems. But what do we learn from this? We learned that if you want to guide a microbial community, you have to know who's there. What are the bacteria there? And you have to know, what are they eating? Because the bacteria that will flourish are the ones that have a food supply. And only those bacteria will flourish. And you have to understand, what are those bacteria that are being fed making? What are the products of those? Are those protective metabolites, nourishing vitamins, or are they endotoxins? So you have to understand that system. And now we do. So we've been looking at the sorts of things that this bacteria does for babies. There's lots of things. It's overtly anti-inflammatory, it's overtly protective. But one of the things we were most concerned with was, if it's so protective, is it possible that the babies aren't educating their immune system correctly? That they're not able to respond to a threat? So we were able to test that. So with Charles Stevenson, he looked at a large cohort of babies in Bangladesh, and most of them were completely dominated by Bifidobacteria infantis, but about 15% did not. And because he was doing a trial on vaccination response, we could ask the question, do you respond better as a baby to a vaccination if you have the right bacteria or the wrong bacteria? And it turns out that if you have Bifidobacteria infantis, you get a much better response to vaccination. You get more antibodies and more diversity of antibodies. So not only does this bacteria protect the baby, it's educating the immune system as it goes. So we're very excited about this result. Turns out, we said, well, who would take advantage of this? Well, one thing, breastfeed, breastfeed, breastfeed. There are huge advantages, and this is one of the benefits of breastfeeding, providing these oligosaccharides. But actually, there's a customer for this discovery that's quite new. Premature babies. Up until recently, premature babies didn't survive. But now modern medicine can keep them alive. Mark Underwood is head of neonatology at the University of California, Davis Medical Center. And his life is, babies are born premature, typically by cesarean section, which means they're born sterile. He takes those sterile babies, He puts them in an incubator and then tries to keep them alive. These babies are in the incubator and they're beginning to acquire bacteria that will populate inside and outside. And where are those bacteria coming from? The hospital. This is not the way you'd like to start life. So we tested. If these babies are getting breast milk oligosaccharides and they get the right bacteria, do these bacteria multiply just like they were a full-term baby? Yes, they do. So now, in effect, we have a solution for Mark so that these babies can acquire the right bacteria. So, given that, we were hoping that a company would, in essence, take this on and productize the bacteria for the NICUs. They all said there wasn't a big enough market, that there just wasn't enough babies. So we actually, as a group of faculty in the University of California, launched a product to do this, provide this product for NICUs. Actually, cesarean section born full-term babies also need the bacteria, so we tested that. We're studying a variety of things associated with that. One thing that's actually rather interesting from a chemical perspective, so this is what's coming out of a baby that's being breastfed. So in the top, that baby didn't have the bacteria, and the oligosaccharides were coming out, water was coming out, the pH was pH 6. They were very watery stools, and the babies were basically making 4 to 6 diapers a day. As soon as they got the right bacteria, the oligosaccharides stopped coming out. Much less water. Actually, they made acetate and lactate, and the pH dropped to pH 4.5. So fewer diapers and pH 4.5. Those of you who've studied microbiology knows that pH 4.5 is an important number. Pathogenic bacteria don't grow at pH 4.5. This is a shelf-stable baby. So we're now realizing that, in essence, this is a very, in essence, protective strategy. Turns out we should have just looked at the old literature. Turns out in 1926 they recognized that as soon as babies started breastfeeding, the pH of their diaper started to go down to 4.5. But over the past almost 100 years, the pH of breastfed babies has been going progressively up. Why? Because we've been killing off the bacteria. Antibiotics, cesarean sections, infant formula, hygiene have lost the bacteria from, in essence, modern Western babies. All right. Turns out it's not just human babies. John Madigan runs the foaling barns at the University of California, Davis Veterinary School. One of the foaling barns that he looks after, 2 years ago in the spring, the first foal of the year, came down with diarrhea on the first day. By the second day, it had died. Second foal, diarrhea. Third foal, diarrhea. They were all getting diarrhea. By the fifth foal, they knew it was an antibiotic-resistant Clostridium difficile infection, and it was now epidemic. Every foal was getting this. C. diff is a huge problem. It's infectious for basically unprotected organisms. So the foals were being born basically sterile. They didn't have a bacterial protection. C. diff is a nightmare. C. diff is a spore former, so the barns are full of C. diff spores. Every foal was getting the problem. So we developed a product, and after 15 foals, we finally got the product into the 16th foal, no problem. Protected them from C. diff in every foal since. So, in fact, we then launched a product so baby horses can be protected from this. So, it's clear that as we begin to understand the way biology works and the complexity of diet and health, we've recognized a whole new target of health, the bacteria within you. So we're basically going to have to eat more food for bacteria and more bacteria. That sort of sounds horrible, but it's not. If you look at the great culinary successes in human history, chocolate, coffee, wine, beer, cheese, bread, yogurt, they're not commodities. They're a combination of a commodity plus a microorganism. And that combination makes the food more safe, more nourishing, more stable, and more delicious. So by mastering this dimension of our health, our food supply will get safer, more nourishing, more stable, and more delicious. So, integration. How are we going to take all of this complexity and start building it into diets? So remember what we've learned just about the bacteria in us. How does that ecosystem work? You have to have the right bacteria, you have to have the right foods for those bacteria, and you have to have, in essence, control on what the metabolites are that they're making. So we know what that is in babies. We, with Carlito, have mapped every single oligosaccharide, every sugar and every linkage in the oligosaccharides in human milk. And that's what babies eat. We've simultaneously mapped the genomes of the bacteria that digest those oligosaccharides and mapped the metabolism onto the metabolites that they make, acetate, lactate, vitamins. So for babies, we're done. All right, a magnificent achievement. Now we can understand what we'll need for adults. If we're going to do this for adults, then we're going to have to be able to map every single polysaccharide in the diet, sugar by sugar, stereospecific linkage by stereospecific linkage. And we're going to have to map the bacteria that you should have in your intestine if you're eating those foods, so we can map those bacteria to the genes they need and the metabolic pathways they have to make that beneficial. So, how are we doing? Carlito has been working for 20 years to develop the technologies he can now do it in high throughput. We've already done 200 foods. By the end of the year, we'll have 500. We have, in practice, mapped the food supply that's abundant. Dave Mills has been building an annotated genomic library of the microorganisms mapped to all of those, in essence, glycans. So we already have what we need. So in effect, can you imagine a day when you have, just like Google Maps, maps of the roads, the highways, and then personally dictating where you'd like to go? Yes, that's what we can do now. And Dave